So quick little review that we pretended like this enzyme was an enzyme that's used to break down glucose, and that we only want this enzyme in the active form when we need to break down glucose and make ATP, and we want it in the inactive form when you already have enough ATP. If you already have enough ATP, then you don't need to break down any more glucose to provide energy to make it. So therefore, those are the two scenarios that we were talking about yesterday. So in the active form, if you are active and are using a lot of ATP, then what we want to do is increase the rate at which glucose is broken down so that we can make more ATP. So you would want this enzyme in the active form. And so we talked about how ADP acts as an activator, because if you're breaking down a lot of ATP, you break it down into ADP. And if we're not making it and regenerating ATP, we start to get an accumulation of ADP, and it activates or binds to the allosteric site, that regulatory site, to stabilize your enzyme to break down glucose in the active form. On the flip side, we were just starting to talk about if, let's say, now you go and lay on the couch and go to sleep, now you're not using as much ATP, and you definitely then don't need to make ATP at the fast rate that you were making it when you were exercising, let's say. And so we want to slow down the production of ATP. How do we slow down the production of ATP? We slow down breaking glucose down. So therefore, we won't have the energy to make any ATP. All right, so that's where we left off. The ATP is going to accumulate. So we're going to add here another bullet here. So what we need to do is stop making ATP because we already have enough. So how do we stop ATP? What provides the energy to make ATP? Breaking down glucose. So we need to stop breaking down glucose. That's cellular respiration. We need to stop breaking down glucose for energy to make ATP. Why do we have to do that? We already have enough ATP. So we don't need any more, so we need to stop it. So what happens is, if your cell is still in high gear making ATP, but you're not using it as much, ATP accumulates. That's why we said here ATP will accumulate. So what ATP does as it accumulates is it acts as an inhibitor. That's what the second picture is talking about here. So here is, so we want it to be in the inactive form. We want to stabilize it in the inactive form so this enzyme doesn't break down glucose anymore. So your inhibitor right here is ATP in this example. ATP is your inhibitor. Notice here the inhibitor binds to the same site, the regulatory site here. So let's write what's going on here. So ATP binds and changes the shape of the active site. Active site. So now this enzyme is in a form that cannot be used. So therefore, can we break down glucose? No. All right, and so I'm going to put here, and cannot break down glucose. So when you're reading it later on. And so therefore, when you have enough ATP, ATP actually shuts down its own production by making this enzyme that's used to break down glucose in the inactive form, and so therefore it won't make any more ATP. Then as your cells, then let's say you get up from your nap and you start using more ATP, your ATP is going to start start breaking down, and that ATP, as you use it, actually can come off of here, and you can use the ATP for energy, and now ADP is going to accumulate again, and the ADP will bind to the enzyme and stabilize it in the active form, and then you'll start making ATP again until you don't need it anymore, and so therefore that's why it oscillates back and forth between the active and inactive form, okay? And so that is called allosteric regulation. So that is one way that our cells control reactions and whether or not they're going to occur or not, because it's real inefficient for all reactions to occur all the time. So this is one way, this allosteric regulation to control that. All right? All right, so let's go back to the PowerPoint and look at, so if you turn your page over, you have this picture here, and I want to point out a couple slides ago when we wrote down here allosteric regulation, we said that the molecule, the enzyme can be stabilized by an activator, so that was like your ADP. We said that it can be, the inactive form can be stabilized by your inhibitor, that was like your ATP. And then we wrote this word cooperativity, and this next picture talks about this, so that's why I wanted to just look at this definition real quick. It says one substrate triggers a shape change in other active sites, and it increases catalytic activity. So let's look at that. So that's the definition of it. So this is showing you cooperativity. And so here we have, here's your substrate, and what happens is the substrate binds to the active site. So this right here is your active site. If the one substrate binds to the active site, what it does is, remember this has 1, 2, 3, 4 subunits, and so therefore has 4 active sites. All it takes is one substrate to bind to one active site to stabilize this whole enzyme in the active form. Meaning that if you have a substrate bound here, you can't have an inhibitor come bind to here. So the inhibitor can't bind there and make it in the inactive form. So this one substrate stabilizes the other 3 active sites so that a substrate can come and bind into each one of those other active sites because the active sites are in the right shape. So they're working in cooperativity, they're being cooperative with one another, meaning that the one substrate bound to that active site is going to trigger these next 3 active sites to stay in the right shape so that the substrate can bind.
And what did you say about the in-between part?
Oh, this in-between part. So remember what this previous picture, that an inhibitor can bind to the in-between part and cause the shape change to happen, yeah. But that cannot happen if there's already a substrate in one of the spots. Okay? So if the substrate gets there first, then it stabilizes this in the active form. And that's called cooperativity. And then the last thing about how we control the rate of reactions with enzymes is this thing called feedback inhibition. So the end product of a metabolic pathway shuts down the pathway by binding to the allosteric site, or that site away from the active site. The purpose of this is to prevent wasteful chemical resources and to increase the efficiency of the cell. Did I go too fast? Sorry. So this picture illustrates this. So let's just look at this. This is a metabolic pathway. Remember I said that oftentimes we look at, here's the reactants, here's the products, but the arrow to convert the reactants to the products has many steps. Usually, that's what this is showing you here. So here is your initial reactant, threonine, and an enzyme catalyzes the reaction to convert threonine into some other chemical, they're just calling it intermediate A. Then intermediate A gets converted to intermediate B by a second enzyme, and then a third enzyme, and so on and so forth. And this is the true end product that's going to be used by the cell at the end of this pathway, isoleucine. And so that's your end product. So therefore, you have to have 5 enzymes working for these this reaction of threonine being converted to isoleucine. So what happens is that while you're making isoleucine is going to be used by the cell for some reason, all right? And so this is used by the cell. So oftentimes you get to the end of the reaction, isoleucine is not going to hang out in the cell, it's going to be used for something. And so, isoleucine is actually an amino acid, so it could be used to build proteins and so on. And so, so this is going to be used by the cell. But what happens is that what if the cell doesn't need it anymore? It has enough isoleucine. What's going to happen to isoleucine? It's going to build up in the cell, right? It's going to kind of hang out. So you're building isoleucine, kind of like you're cooking dinner, all right, or cooking for people, and you keep on making this food, but when all the people are full, all right, if you keep on making the food, you get this buildup of food and nobody's eating the food, and then it just kind of hangs out and it's wasteful. That's the same thing in the cell. So they're making isoleucine. In my analogy, isoleucine is the food. Usually it's used by the cell, all right, but when the cell doesn't need it anymore, it starts to build up. So if this builds up, this is what happens here, this red line here. If it builds up, it actually, this end product actually acts as and stops this enzyme in the very first reaction from working. So how does it do that? Notice here the isoleucine can bind to that enzyme, so that's that enzyme here, at a site that's different than the active site. So that's an allosteric site, so isoleucine binds to the allosteric site. And notice that when it does that, what did it do? This is the enzyme here. The enzyme is supposed to look like this. The active site is now changed. The shape is changed as a result of that binding to the allosteric site. So it's acting as kind of an inhibitor of this enzyme, and this enzyme won't work. And if this enzyme won't work, will you make any more isoleucine? No, because if this enzyme, every enzyme has to work here in order to get the isoleucine. So if we stop it at the first step here and it acts as an inhibitor to that enzyme, then that first reaction doesn't happen, which means none of the following reactions happen. And then, so this happens when isoleucine, I'm going to put here, isoleucine builds up in the cell. Or that means it's not being used. And it's in that situation that it inhibits this enzyme. If it's being used by the cell, it's going to be used right away, and we don't get this pathway going on. So it'll keep on making isoleucine, and all the enzymes will work and be used by the cell. But if it's not, it builds up and inhibits the enzyme in the first spot. And this is reversible though, because what if, so the isoleucine builds up in the cell, it's not being used, but then what if you come at a point in time where it is being used again? So if it is being used again, that's what this is showing you here, isoleucine used up by the cell. So when it starts to be used again, it actually can come off the enzyme and be used. The enzyme goes back to its normal shape, and then it continues to go and make new isoleucine. And so, so therefore it kind of fluctuates back and forth between those. Let me ask you this, why is it beneficial for the end product to inhibit the very first enzyme in this pathway? Why doesn't isoleucine inhibit enzyme 4? What's the benefit for affecting that first enzyme in the pathway? Megan?
Would there be the buildup of intermediate B if you stopped it like at enzyme 4, and then there's no way to like start that back up again? Like there wouldn't be a reason to have a built intermediate B or
Yeah, there wouldn't be, if we stopped it at one of these other enzymes here, like if I denatured one of these enzymes, you'd have, you're right, you'd make these intermediates and you'd have a buildup of them because they wouldn't go anywhere further in the reaction. And really, like intermediate C or intermediate D or any of these intermediates are just to get to your end product. So do you see where it would be pretty wasteful to go through half the reaction and not get your end product? All right. So therefore, it'd be like it'd be pretty wasteful for me to go through half of making the meal and then not finishing it up. All right. That would be wasteful. I would have wasted all my ingredients and things like that. The same thing with this is that if you go halfway through and don't get your end product, all of that was wasteful. And so therefore, if you're not going to make, if you don't need your end product, it's more efficient for the cell to never have this pathway to begin with happen. And so therefore, the first step, you don't even start making the meal, all right, if there's nobody to eat it. And so in this case here, the first reaction, if you stop that, then you never make any of these intermediates and there's no waste, okay? And so that is feedback inhibition. And so I'm introducing this to you now. We come into contact with this in other areas of our curriculum later on. All right, and that is the end of the notes. Okay, so what I'd like you to do is turn your attention to the sheet you picked up today. Because what we're going to do is simulate some things with enzymes today. I'd like you to look at that sheet and read the first page. Guys, get to be an enzyme today. Some of your dreams are coming true. So read that first page. Okay, so let's just talk a little bit about this. So you, as I said, you're gonna count out 40 toothpicks, and so I have little boxes of toothpicks, and so what I'll do is just put like one per row and we can pass it back. You're gonna work in partners and pairs, and so there are even numbers of people in the class, so that works out perfectly today. And so you'll count them, and it says place them in your bowl, so you also get a paper bowl. And you are going to act as the enzyme, as it says here that your hand is the enzyme. You're going to be working with a partner, so you have to decide between the two of you. One of you is going to be the timer, one of you is going to be the enzyme. I'd like the enzyme to stay the enzyme for the whole activity because there's two different trials. Why is it that I wouldn't want you to switch? Michael?
One person might be better, and then now you have different things.
Yeah, so you're introducing a different variable about why your results may be different. So stick with the enzyme that you pick. So and the other person is going to use the timer. For this activity, I'm going to let you use your cell phones for timers. Okay, so after we go through it, you can come up and if your cell phone is up here, you can come up and get it. All right. So and then your hand is the enzyme, and you're supposed to use your thumb and your fingers to break it. Okay. So it's not like you're picking up out of the bowl and breaking it like this. All right. Picking one hand and you're not looking because can enzymes see? No, they cannot. So your bowl is like the cell. The toothpicks are like the substrates in the cell, and so you are the enzyme, and the enzyme picks up a substrate and is going to convert it to product. Your toothpick, the broken toothpick, the two sides that are the two halves are going to be your product. So just like in a cell, when a reaction occurs, the product stays in the cell. So after you break it, you put the two broken parts, the product back in the cell. It's going to stay in the cell. Then as the enzyme, you go and try and find another substrate, break it, put it back, substrate, break it, and so on and so forth. And so you keep putting it back in there. It tells you to do 10 seconds at first. So the timer is going to do 10 seconds. The person's not going to be looking, and they're going to break as many. You want to be as best of an enzyme as you can be, all right? But what you don't want to do is you don't want to break the products. So if you need to feel for the longer toothpicks and not break a product, which is going to be the shorter broken in half toothpicks, all right? So you want the longer toothpicks. After 10 seconds, you're going to stop, and then you're going to look, and you're going to count how many toothpicks you broke. And remember, 2 halves equals 1 whole, all right? 1 toothpick broken. So when you look at the back side of the first page there's a data table. And so there's time, we're measuring it in seconds, and then there's a column for trial 1. So this is what I'm describing as trial 1. So notice here at time 0. At time 0, when you're writing the number of toothpicks broken or metabolized, what should you put it, everybody at time 0? 0, right? So everybody right now put 0 there. And then, so then you're going to put however many you broke in 10 seconds. Then our next one is 30 seconds. So what you're going to do, instead of starting over, you're going to put all your broken toothpicks back in. That's how many you broke in 10 seconds, and the timer is going to time you for an additional 30 seconds. You're going to try and break as many as possible for that additional 20 seconds. Then stop it. And then how many you have broken there is what you would have had in 30 seconds. Does that make sense? 10 seconds for the first trial and additional 20 seconds. And you're going to follow that logic for 60 seconds, then 120, and so on and so forth. And you're going to, so at the end, it's cumulative. So, you know, it's how many total broken at the end of that time period. So you're filling out this column. Then if you turn to the next page, there's a graph to do, and so I put here on the x-axis is time. So we already decided the time. You're going from 0 to 360. So you remember you want to use as much of the axis as possible. So 0 on the left, and you want to go all the way, or very close to all the way, to 360 at the other end. What would go on the y-axis? The toothpick's broken, right? And so that would be, in our analogy, that's the substrate being converted to product, how many of that. All right, so you're gonna do a line graph for that. Then if you go back over to the table, there's a column for trial 2. So for trial 2, notice it says number of toothpicks broken or metabolized, and then there's a blank, and it says, blank is the variable that is changed. While you guys are working in pairs on trial 1, I'm going to come around and tell you what you need to do to alter trial number 2. So these are the different, I have a little sheet here that I'm going to write your names, who I assigned who to, what trial, what change that you're going to make. But some people in the class, some groups are going to change the substrate concentration for trial 2. So you're going to do the same thing for trial 2, except you're going to make the change that I assigned to you. And I'm going to leave it up to you how to change that. All right, so you can change the substrate concentration. It could be an increase or a decrease. Some people are gonna, I'll assign that you're gonna change the enzyme concentration. So you have to think about with your partner about how you're gonna do that. Some people I'm gonna assign that you're gonna introduce competitive inhibitors into your bowl. So you have to think about what it means to be a competitive inhibitor, what you can do to simulate that in this toothpick example. And then some people I'm going to assign a denatured toothpickase, so the enzyme becomes denatured. So how can you simulate a denatured enzyme and do this, okay? So you're going to have to be creative and think about how you can adjust this activity to relate or to show these different things. Tristan?
So for the denatured toothpickase, you're not going to break our fingers, right?
I promise I will not break your fingers. No. In fact, you're going to decide. So there's going to be multiple groups that do denatured enzymes. Like last hour, I had 3 groups that did denatured enzymes, and all 3 groups did something different. All right. So you can decide what you want to do. The thing is that what you decide, when you figure out what you want to do, I want you to call me over and just run it by me. All right. So just so I can make sure that it's actually what it's supposed to be. Okay. Question.
So are we doing 40 toothpicks on the first one and then the second one? So we need 80 in all.
Yes. So you're gonna do 40, you guys, toothpicks in the first one, and then to accurately compare, it depends on what you're doing actually for trial number 2. All right, as how many that you need. So we're gonna do trial number 1 first, and then we're gonna work on trial number 2 because right now you don't even know what you're gonna be assigned. Okay, so let's get our toothpicks. So I have some boxes. The cashier looked at me like I was crazy when I bought 5,000 toothpicks. We're like, yeah, 5,000 toothpicks.
Made off a steak.
Luckily they're small, there's a box, so. I can open this box too. So, if they're not open, they open, it says open here at the end. All right, so you can get your 40 and pass the box on down the row. And so when I have another box here, so we can start. And then how about one person from every group come up and get a bowl?
Yeah, I'll get a bowl with my throat.
And you guys want to work together? All right, let me stop the recording.